Hello, I'm Stine Østerhus. I'm a PhD student at the Norwegian University of Science and Technology. And my PhD is a part of a larger project called Task Force Salmon Lice. And here are the partners to the project. It's mainly salmon-producing companies, technology providers, and research institutes. And today I will talk about some of the research I've been doing. It's called Energy Resources and Decline During the Free-Living Stages of Sea Lice. It's a bit different than the title in the program, but the content is the same. And this research was done in collaboration with another PhD student at our project, Maria Guttu. And Cecilie Miljeteg has done a lot of the practical work and the video analysis, and Anna Solvang, she's a researcher at our project. And Anna Solvang Båtnes has also helped out a lot with the practical work. And she's also a researcher, but the project coordinator of Task Force Salmon Lice as well. And Dag Altin has been helping a lot with the construction of the setup and brainstorming throughout the experiments. And Ingvar Olsen and Atle M. Bones are my supervisors. So first, I will just give an outline of what I will be talking about today. First, I will take some general background for the experiments and the setup, which is mainly the rearing conditions for the lice we've been using. And we've done like 2 sets of experiments in parallel or simultaneously. We've looked at vertical swimming behavior and decline in carbon content. So I will present the methods, specific setup, results, and do a summary for both of them. We've been working with 2 species of sea lice, Caligus elongatus and Lepeopteris salmonis. And in our lab, we were cultivating 4 strains of sea lice, 3 Lepeopteris salmonis strains, LS gulen, LS wild, and LS sulfur, and 1 Caligus elongatus strain. The LS gulen strain has been cultivated for many generations at Bergen and for some generations at our lab. So it has probably been reared around 80 generations in the lab at the time of these experiments, while LS wild and CE and LS sulfur have been reared in the lab for 4 to 6 generations upon these experiments. And the aim of this research was to compare the energy levels at specific time points after hatching, because data regarding their energy consumption can provide valuable input data for spread risk models, fallow practices, and it can provide lots of valuable data. And we looked at the vertical swimming behavior and carbon content of the lice as measurements of their energy. As upon hatching, these lice are non-feeding. They depend on their endogenous energy supply, which is a lipid droplet within the louse. And lipids are very carbon-rich. So this is our general setup, or the rearing conditions. We had styrofoam boxes filled with water, which contained an incubator, which in turn contained incubator pipes, where egg strings were hatched. They were kept at 10 degrees Celsius under constant light conditions and had a flow of water through the incubator pipes of 3 milliliters per minute. And we took samples at specific time points. So for our vertical swimming behavior experiments, we took samples at 2, 5, and 12 days post-hatching. And when we sampled, we took 10 larvae and put them in a clear plastic cuvette and filmed them. 3 replicates were done per sample, and the video files were analyzed using EthoVision. Just a quick disclaimer, we're still very early in the process of both analyzing the data, analyzing the videos, and processing the data, so I will only be presenting some preliminary results from the LS gulen. Oops. Sorry. Okay, so this is the specific setup for the vertical swimming behavior experiments. The cuvette was placed on a platform with a black background, and we had light sources from the sides and above. And otherwise the room was dark. Upon placing the lice in the cuvette, we put them on the platform and allowed for them to have a 5-minute resting period to adjust to their new conditions before we filmed them for 2 minutes. And here you were supposed to see a video of the lice moving around, but the PowerPoint became too big, so you just have to imagine them swimming around. But you see it like you see these like oblong white dots. I'm shaking, so I hope you see them because I can't point. Those are the lice swimming around. And on the right here you have the tracks that were detected in ethovision, and each colored line represent the individual or the tracks of one individual louse. Over to the results. The orange diamonds here, they are not regarded, they are only to show how big percentage of the lice in the sample were actually moving, because not all lice were detected and not all of them were moving. So it will be the same for all the plots I'm showing. It's just, do not pay too much attention to them. It's just to show there's a difference. And not all were moving. But here on the left, you see the mean velocity of the lice at in millimeters per second at the different ages. And you can see that they had a higher mean velocity at 2 and 12 days than on 5 days. But none of them really had a very high mean velocity, and that's due to them having periods of time where they're not moving, when they are sinking, because their sinking speed is lower than their active swimming speed. But this was like the pattern. And on the right, you have, the time or the percentage of time that they were actively moving. And to make sure that that only their active movements were detected, we put a lower threshold of movement at 1.5 millimeters per second, because their sinking velocity is around 1 millimeter per second. So we put a lower threshold for it to be detected as movement. And here you see the same tendency as for the mean velocity, that age 2, when they're at 2 days old and 12 days old, they are moving a bigger percentage of the time than when they are 5 days old. So this indicates that the lice are more active at the early and late stages than in the middle. And here we also divided the cuvette into zones, with zone 1 being the top zone, zone 2 being the middle zone, and zone 3 being the bottom zone. And on the left, you see how much time it took for them to get to different zones for the first time. How many seconds did they spend before reaching the different zones for the first time? And as you see, they all spent the most time reaching the top zone, zone 1. But at day 12, they spent less time getting there than at day 2 and day 5. And on the right here, you see how big percentage of the time they spent in each of the 3 zones. And they all spent the most time in zone 1. And especially at 12 days, they spent a much bigger percentage of their time in zone 1 than zone 2 and 3. So it seems as though the lice spend more time getting to the top zone, but they prefer to stay there. So summary, yeah, we are early in the process. The data needs more work, and 2 of the LS strains and Caligus elongatus strain is not yet analyzed, so it will be very interesting to process those data and see, are there any differences between the species and the different strains. The early and late stage lice appear to spend more time moving and move faster than the middle stage. And although they spent more time getting there, all ages preferred to stay in the top zone. And now over to our other experiments, the decline in carbon content. Here samples were taken at 2, 5, 8, and 12 days, 13 for the Caligus elongatus post-hatching. There were 150 larvae in each sample, and we did 3 replicates per sample. So what we did was we picked 150 lice using a pipette, put them in a petri dish with a fine mesh bottom, dried them off, and put them into tin ships and used combustion analysis on elementar vario L-cube to analyze the carbon content in the sample. And for the data processing, we used amount of carbon per individual to calculate. And here you see our results. LS gulen is blue, LS wild is orange, LS sulfur is gray, and CE sulfur is yellow. But you can see that the samples for the Caligus elongatus look a bit strange with them having this increase up until day 5 and then having a decrease. And you can kind of see the same tendency for LS sulfur with this flattening till day 5. And we know why. It was because we rinsed the lice with deionized water before picking the samples, and this caused the lice to burst and thereby providing unreliable data. So for the nuclear stages, so we chose to look at them from just the day 5 samples when they were all copepodites. And here you can see, not surprisingly enough, the non-feeding lice using their endogenous energy supply has a decline in carbon content. And you can see that the Lepeopterus salmonis strains, they have quite a similar both starting amount of carbon, it's in micrograms per individual, and decline rate. The carbon content was between 2.1 and 2.5 micrograms per individual, while the decline rate was at 0.14 to 0.18 micrograms per individual per day. But you see that the Caligus elongatus, it has a lower starting concentration and a lower decline rate than the Lepeopterus salmonis. It has a starting concentration of approximately 1.3 micrograms per individual and a decline rate of 0.07 micrograms per individual per day. But as you know, the Caligus elongatus, they are a bit smaller than Lepeopterus salmonis. Even in the early stages, they are at least not as big a difference as the adult stages, but the Caligus elongatus are smaller. So that could account for the lower starting concentration and also the lower decline rate, because even though they are using or spending a smaller amount of carbon per day, it could be a bigger portion of their starting concentration. To see if this was the case for the decline rate, we looked at the carbon loss in percentage. And here you see the same tendency with LS gulen being having a steeper decline rate than the others, and that the CS sulfur still has a lower decline rate than the Lepeopterus salmonis strains. At 12 days, the Lepeopterus salmonis strains were had between 45 to 50% of their initial carbon content at 5 days, while CE sulfur, Caligus elongatus, had 53% of the starting carbon content at 13 days, so it had a bigger percentage left at a day later. And this is just not really related to that in any other way than at 10 degrees, the lice will hatch at different times. It could vary between the 0 days, the day of picking the egg strings, to up to 9 or 10 days. And we wanted just to see if there was any obvious connection between hatching time and the carbon content of the lice. And as you can see here, the different colors are different. Here is different species. On the next slide is the 2 different strains. And the darker the dots, the earlier the egg strings hatch, and at lighter the dots, the later they hatched. So as you can see, sometimes the ones that hatched late had a higher content than the ones that hatched early. Other times, the ones that hatched on the same day had quite a big variation. And sometimes the ones that hatched early had a higher concentration than the ones that hatched late. But still, this is, so it seems like they're a bit all over the place, and can't really see any obvious connection between hatching time and carbon content. But there's not enough data, and these data were not meant to do this. Then we would have had more sampling times, and I just wanted to see if there was an obvious connection, and there doesn't seem to be one. So over to the summary. Caligus elongatus had the lowest initial carbon content and the lowest decline rate, while LS gulen had the highest initial carbon content, but also the steepest decline rate. But all the salmon lice strains were quite similar in both carbon content and decline. And the hatching time does not appear to affect the carbon content. And here you see a Lepeopterus salmonis naupli on the bottom, and Caligus elongatus naupli at the top, just to see. It's quite a difference. Thank you so much for listening to me. I'm sorry for messing up a bit.